So first, let's look at everything we need to know about taste buds, beginning with their location. So most of the taste buds are present in the papilla of the tongue. So they are present in all the papilla except for the filiform papilla. So right now we're only concerned with the fungiform, foliate, and the circumvallate papilla. The fungiform are present mainly on the anterior surface, the foliate on the lateral margins posteriorly, but the foliate papilla aren't well developed in humans. And you have the circumvallate papilla, which are present in a V shape on the posterior surface of the tongue. So besides the papilla, taste buds are also present in the palate and the esophagus. And one more thing you should remember is that there are many thousands of taste buds in the adults, but beyond the age of 45 years, the taste buds begin to degenerate, and this reduces taste sensitivity in older people. So here's a nice diagram showing the structure of a taste bud. You can see that's composed of the receptor cells, which are actually sensory neurons, and the surrounding supporting cells. The taste receptor cells have microvilli, which are also called the gustatory hairs, and the cell membranes of these hairs, they contain the receptor proteins which actually bind to the chemical substance, and after that action potentials are generated, but we'll talk about that later. And at the bottom you can see the axons of these sensory neurons, and these axons are stimulated obviously when the taste receptor cell is stimulated. Okay, so now we can say that each taste bud is specific for one primary stimuli when that substance is present in low concentrations. For example, when either a sweet, sour, salty, bitter, or an umami substance is present in low concentrations, it will excite only a single taste bud. However, the taste buds respond to two or more of the primary stimuli when the concentrations of those substances increase. Before we talk about the actual mechanism of how taste cells are stimulated, we should first remember that taste receptors, like all other receptors, can be either metabotropic or they can be ionotropic. Okay, so let's look at this in the context of taste buds. So when a chemical substance binds to an ionotropic receptor, this receptor will cause the opening of an ion channel, and sodium ions will diffuse inwards through this channel. And if the chemical substance activates a metabotropic receptor, the receptor would activate a G protein, and then eventually cyclic AMP would be produced or some other kind of second messenger, and this second messenger would result in the opening of the same sodium ion channel. So these channels can be opened in two ways, either directly by the stimulus or you have indirectly through the G protein. So the salty and sour stimuli, they work mainly through the first mechanism, which is by opening sodium ion channels directly. And then you have sweet and bitter, which work mainly through the second mechanism involving G proteins. So most of the chemical substances excite the taste cells through the first mechanism. So that is that they bind to the receptor, the taste receptor, which is present in the cell membrane of the taste hairs, and this causes the opening of sodium ion channels or the movement of hydrogen ions. And these sodium or hydrogen ion channels, once they enter the cell, they cause depolarization, and the membrane potential becomes less negative, and this in turn causes the opening of voltage-gated calcium ion channels. The entry of calcium causes the movement of vesicles containing neurotransmitters towards the presynaptic membrane, and the release of neurotransmitters then excites the axon of the sensory neuron. So let's look at the primary sensations of taste. So like the primary colors, you also have primary taste sensations, and these are sour, salty, sweet, bitter, and umami. So let's look at how these taste sensations are caused. You have sour taste, which is obviously caused by acids and is related to the hydrogen ion concentration. So the greater the hydrogen ion concentration, the more sour that substance feels. The second is salty, which is caused mainly by ionized salts such as those of sodium chloride, and it's basically the sodium ion concentration which is basically responsible for the salty taste. Then you have sweet taste, which is caused by many organic compounds like sugars such as the monosaccharides and disaccharides, the aldehydes, ketones, alcohols, inorganic salts of lead and beryllium, and many other compounds. Then you have bitter taste, which is caused by long-chain organic compounds containing nitrogen, as well as alkaloids. And the alkaloids include substances like caffeine, which is present in coffee, and then quinine, strychnine, nicotine. Also, some substances initially they taste sweet, such as saccharin, but after that they become bitter. So any bitter taste indicates the presence of toxic substances or poisons in the food, and this has an important function because it causes the person or the animal to reject that food. So that's why you can say that bitter taste serves a protective function. And lastly, you have umami, which basically means delicious, and this taste sensation is different from the other four taste sensations, and it tastes nothing like them. So this taste is experienced in foods containing monosodium glutamate, which is actually present in Chinese food, as well as meat extracts and aging cheese. However, many people are intolerant to monosodium glutamate, and because of this, they experience Chinese restaurant syndrome. So the threshold for taste basically means the minimum concentration of a certain substance that we need to stimulate a specific taste. For example, to stimulate the sour taste, you need a minimum concentration of 0.0009 molar of HCl. And just like that, there are thresholds for sweet taste as well as for bitter taste. Okay, so the important thing to remember here is that bitter taste has the least threshold. For example, you need a minimum concentration of 0.000008 molar of quinine to stimulate the bitter taste. This basically means that you need such a low concentration to stimulate bitter taste. So why is that important? Because bitter taste is most sensitive compared to the others. It provides an important protective function, and it informs the person or the animal that there are there are toxins present in the food, or there could be poison. So this causes the person to reject that food. So in this table, you can see relative taste indices. So you have sour substances, bitter, sweet, and salty. So in sour, HCl is taken as 1, and quinine as 1 for bitter substances, sucrose for sweet substances, and NaCl for salty substances. So all of them are taken as an index of 1, and the other substances are compared to them. For example, HCl is the most sensitive, and carbonic acid is the least sensitive. The substances at the bottom have a very low taste index, and because of this, large concentrations of these substances are required to stimulate that taste. Now let's look at the transmission of taste. Taste sensations are carried by 3 nerves, which are the facial nerve, the glossopharyngeal nerve, and the vagus nerve. Taste sensations from the anterior 2/3 of the tongue, they're carried by the lingual nerve, which joins the chordae tympani branch of the facial nerve, which eventually joins the facial nerve itself. Taste sensations from the posterior part of the tongue, it's carried by the glossopharyngeal nerve, and taste sensations from the base of the tongue, as well as the parts of the pharynx and the epiglottis, are carried by the vagus nerve. So the first order neurons run in the ganglia of the facial nerve, the glossopharyngeal nerve, and the vagus nerve, and the second order neurons are present in the nucleus of the tractus solitarius and the medulla. Then from the nucleus of the tractus solitarius, the axon synapse for third order neurons in the ventral posteromedial nucleus of the thalamus, and then from the thalamus, the axons that project go to the taste center in the cerebral cortex. The taste center is located in the lower part of the postcentral gyrus, which is concerned with sensations coming from the tongue and other parts of the mouth. And it's also present in the opercular cortex in the insula. This is the insula, and as well as part of the lateral sulcus. So this pathway functions to provide conscious perception of taste. So if we talk about taste reflexes, then the taste reflexes involve the nucleus of the tractus solitarius, which sends its signals to the salivary nuclei, the superior salivary nucleus of the facial nerve, and the inferior salivary nucleus of the glossopharyngeal nerve. So signals in the salivary nuclei then obviously innervate the salivary glands and control the secretion of saliva. Another feature of taste sensation is that it's a rapidly adapting sensation. For example, you eat some kind of food, for example, a sweet food, and the first time you eat it, eventually you'll become adapted to it and it won't feel as sweet as it was the first time that you ate it. And if you eat something really sour, the first time you put it in your mouth, it may seem so sour to you and you won't like it at all. But as you keep chewing on that food, eventually you'll become adapted to it and it won't feel as sour to you as it did the first time. So half this adaptation occurs in the taste buds themselves, and the other half takes place in the CNS. And because of this reason, taste adaptation is a little bit different than adaptation of other receptors, because for those receptors, adaptation occurs completely at the receptors and doesn't involve the brain.